I have had the pleasure of working with Dr. Garzon and Dr. Georgia Napolo, or Pam and Demetra, from the beginning of women's venture competition. They're my mentors. They advise me, my venture, but they more so, they are drug developers themselves. They are scientists. They give time very, very generously to other women, to leaders, to entrepreneurs. They are investors themselves. They are entrepreneurs themselves. I'm just so grateful to both of you and for our community. So with that, I will give you Pam and Demetra.
So I feel privileged, and I am always humbled to collaborate with the esteemed scientists, the clinicians, and the investors on the Aim High Women's Competition Committee. My co-chair, Dr. Raju Kuchalapati, Dr. Webb Cavanagh, and Dr. Brian Leon Jones in particular. The competition, as you heard, is 4 years old. And since that inaugural event, this committee has reviewed 215 applications. These applications came in from over 20 different countries worldwide. We listened to 30 pitches, and each of those pitches were followed by a question and answer session. Some of the questions were more insightful than others, but nonetheless, we sat through it all. And we recognized 12 of the participants, either through an honorarium, through endorsement, or in the seed funding. Just this past year alone, we received 71 different applications from 6 countries. And they represented different areas. We had 33 applicants that dealt with therapeutics. We had 19 that were in the diagnostic field. We had 8 that were in digital health. And we had 11 that were in this category technology that included imaging and detection modalities, as well as drug delivery aspects. So very diverse group. And I think, and Sujan talked about this, but what I think is so unique with the Aim High organization and what Sujan has built is that the participants get advice, they get mentoring, they get coaching, and Sujan is fantastic at enabling connections across the board in different areas. And I applaud her and the team for enabling this effort. And it is really great to see this wonderful community of innovative, creative, passionate women entrepreneurs to avail them of these resources to allow them to succeed in what is sometimes a very highly competitive field. So thank you so much. It's been my privilege to work with you. And for now, I will turn it over to Demetra to introduce our awardees.
Thanks, Pam and Rose and Sujuan and the whole committee. Yeah, it's my pleasure to introduce you to the two finalists, first award and second award. The first one is Sarah Hein from March Biosciences. I have intersected with Sarah in 2023 at almost every investment event there is out there. I saw her multiple times throughout the year. She was at every fundraising investment event, and I was very impressed with her. She's one of those quiet powers that comes out of the flyover states, you know who they are. And she's been involved with multiple successful biotech companies, as well as a lot of incubators out of Texas. And she's going to be talking to us today about her company with March Biosciences on the late-stage clinical cell therapy technology. And then secondly, the second awardee is Lily Zhu, the CEO of Tegron Therapeutics. I also was very lucky to meet with Lily in a very conspicuous situation at Bio in Boston this year, but we were able to meet and catch up and talk about this event and her work with Tegron Therapeutics. and her work in the highly innovative protein degradation and molecular glue space. So without further ado, I'll invite both Sarah and Lily to come to the stage. Thank you. Oh yeah, I'm sorry. So Joanne, come and present her awardees. Thank you.
All right. Well, thank you all. As I think I'm better at building companies, hopefully, than at coordination, but that's okay. So thank you all, first of all, to the committee, to Sujuan, to Dimitra for having me here today and giving me an opportunity to talk about March Biosciences. It's been an incredible journey, and it's a privilege to be here in front of so many names that I recognize on paper and not in person, so thank you. My name is Sarah Hein. I'm the CEO, co-founder of March Biosciences. Next slide. Do I have a clicker? All right. So March Biosciences is a cell therapy company. We're unusual in that we're new, but we're starting on a clinical stage asset out of Baylor College of Medicine Cell and Gene Therapy Center. I'm going to spend the majority of the time talking about our lead asset, which is targeted to T-cell lymphoma and leukemia. And so, as many of you know here, we've made tremendous strides at treatment for the B-cell cancers, which have a number of targeted therapeutic options available to them today, including targeted therapies, immunotherapies, as well as the traditional chemotherapies, trying to get to those transplants, and the prognosis for those patients is quite good. Unfortunately, the treatment for T-cell cancers is still incredibly poor. And so these patients have a poor prognosis at the start, and unfortunately, once they enter the relapse and refractory state, they have an incredibly poor prognosis and survival of only 10 to 20% over about 3 years. And the reason for that is that the shared target space between your normal T cells and your cancer cells limits your ability to develop those targeted therapeutics that are so important for the treatment of other cancers. And so this is the same case, of course, for CAR T-cell therapies. And then you have the additional complexity with CAR T-cell therapies that if you're using T-cells to target T-cells, of course, you risk that you will not be able to make a product at all. That's slightly more overcomable through engineering advancements that we have today. However, the challenge is if you do those sort of molecular gene editing adaptations, what you're doing is you're adding a lot of regulatory complexity, you're adding a lot of CMC complexity, and ultimately, we think that there's a better way of doing this where we have a strategy through in-depth engineering where we do not have to do that additional gene editing at all. So the lead asset for this product, for this company, we're calling MB105. It's a CD5 targeted CAR T-cell therapy. CD5 is widely expressed on your normal T cells. About 95% of those T cells will express CD5. But you can leverage some natural biology of the CD5 receptor that's unique to that receptor. If you design the right CAR and express it in the right way, you can actually drive internalization of that receptor, allowing you to create a product, and it also protects your normal T cells, or at least some of them. But you still get exquisite efficacy against both the leukemic and the lymphatic cells that are expressing that CD5 receptor. This has been widely characterized, and I know I'm in a room full of scientists, but we're going to skip a lot of that wonderful mechanistic work so we can talk about the clinical data and the results coming out of the clinical study. So there's an ongoing investigator-initiated phase 1 trial in Houston, Texas right now through the Texas Children's Hospital and Houston Methodist, and it treats both lymphatic, lymphoma, and leukemic patients with the T-cell diseases. It's a standard dose escalation trial. What I would draw your attention to actually is that the patients here are very heavily pretreated. And I think somebody commented on that earlier, that it's just impossible to treat patients when they've had 5, 6, 7, up to 16 lines of prior therapy in this trial. And so the other thing I would draw your attention to is that actually this is a outpatient treatment first because of the excellent safety that we've been able to achieve. And so what I'm showing you right here are the initial responses in the lymphoma patients. And you can see the patient here on the scan came into the trial with incredibly heavy disease burden and was cleared of his disease just 4 weeks later. Out of the original cohort of lymphoma patients, what we see is 3 of those achieved a complete response and 1 partial remission. Very favorable safety profile, especially for cell therapy. Low to non-existent cytokine release syndrome, low to non-existent neurotoxicities. So incredibly encouraging. It's not where it is for the B-cell malignancies, but you can see the signal here. And what's interesting is a lot of the patients actually were given product that was produced under what we're calling the shortened manufacturing protocol, where you shorten the expansion phase of the product production. This has been done in other CAR T-cell therapies, tends to be associated with a more favorable response because you're preserving the less differentiated cells. But what's interesting is the only patients responding here are from that protocol. And what we think is happening is that in this product in particular, what you see is that the continual re-expression of CD5 is exacerbating the tonic signaling that you see in a lot of cell therapies. That's driving an excessive amount of differentiation above and beyond what you would typically see, and that's limiting your ability to have a potent product in many of those patients. And so not only can you shorten the manufacturing, you can actually induce inhibitors of that tonic signaling, which can further block it entirely and preserve your cell fitness. This was tested in a mouse model, looked very favorable, and so they put it into the GMP system. And what you're seeing here is that the shift in the preservation of those naive-like T cells from originally about 5 to 10% to upwards of 20-ish% of the product now is those naive-like T cells. And I don't think I have to overemphasize here that the responses that we've seen since then are remarkable. Originally, a lot of these patients, you'll notice, are actually leukemic patients instead of lymphoma patients. Originally, we were not seeing responses at all really in the leukemic patients. We are now sitting at about a 60 to 70% response rate across leukemia and lymphoma, and this is definitively tied to the increased persistence of those CAR T cells. And so this is a phase 1 study. We're still relatively early in the exploration of this, but we do feel confident that we have a very favorable signal here, and we're looking to move this into a phase 2 trial. And one of the things that we're trying to do as we move into that phase 2 trial is really focus on the challenges that have plagued cell therapy for previous companies. And so right now, the majority of the work that we're undertaking is really focusing on the manufacturing of this product, streamlining it, making all of the major changes that we expect to make for the life of the product through our partnership with the CTMC, which is able to move very, very quickly. It's actually a spin-out from MD Anderson after they had reinvented that facility from Bellicum. And that's going to give us a scalable product that then can move into that phase 2 trial and create a consolidating patient data set. The other thing you'll notice is that this trial is focusing on a T-cell lymphoma population very specifically. We do think that there's many advantages to focusing on that. The CD5 expression is the most robust of all the potential T-cell targets that you could have in that population specifically. And it is a significantly larger market and addressable disease for initial labeling of this product. So initially, as I said, we're going to focus on second line and above. That is quite supportable. The need for these patients, I just cannot overemphasize it. The last approval was almost 10 years ago for a specifically labeled product in this patient. There are additional trials. You can almost count them on your hands for how many trials are actually available to these patients today. Then you can expand into ALL, and there's actually a couple of B-cell malignancies that are characterized by their expression of CD5 as well. And these are malignancies that have tended to, over time, relapse after a CD19 CAR T-cell therapy. There is a pipeline in development. We don't have a ton of time to go into details here, but really our goal is not just to use this, to go after this phase 1 product, but also to expand into other indications, other targets. We tend to know what the right targets are for cancer, but there's a lot of challenges with on-tumor, off-target toxicity, off-tumor, on-target toxicity, and we think that there's additional engineering that we can do by paying attention to science, paying attention to biology to unlock those targets. The team that we've been able to put together in just a very short time since our initial funding is frankly nothing short of remarkable, with the kinds of people that we've been able to find, all specialized in cell therapy, typically about 20 years of experience in the field. And I have to say, I wish I would suggest my experience as a leader, but it's really the strength of the data and the excitement around this project that's allowed us to form both this team as well as put together a truly stellar scientific advisory board as we've launched, as well as bring in some highly experienced independent board members as well. And so we're moving very, very quickly. What you'll see is that we actually took on our initial funding in January this year. Everything that we've been doing since then has been focused on CMC, regulatory work, and clinical development work, just trying to bring the company up to this maturity of the asset so we can stay ahead of the opportunity in front of us. We are on target for that Q2 2024 IND to go into a phase 2 study. We are fundraising to do that right now. We've made significant advancements towards those fundraising goals to support that study. But if you have some friends who are interested in a very unique opportunity in cell therapy, please do have them reach out to me. And with that, thank you.
Next is Dr. Zhou.
Good afternoon. I'm humbled and honored to be here, to be one of the winners of the venture competition. I want to thank Sujun and the committee and the Aim High Accelerator Fund for the support and inspiration for women scientists and entrepreneurs worldwide. I'm Lily Zhou, co-founder and CEO of Degron Therapeutics. Degron is a startup company focusing on the discovery and development of a drug called molecular glue degraders. We target the undruggable targets for diseases that have high unmet need. So just a quick overview. Oh, the title doesn't show here. What is a molecular glue degrader? So molecular glue degrader belongs to a new modality called targeted protein degradation drugs. It's a small molecule drug, but comparing to traditional small molecule inhibitors usually bind to an enzyme or a functional protein and inhibit the function of that protein. But targeted protein degradation drugs, whether it's a PROTAC or a molecular glue, they function through the degradation of that target protein. So this system, the drug hijacks the system, proteasome system. It brings the E3 ligase to the target protein and add ubiquitin to the target protein. So the proteasome system would recognize that protein as trash and will then degrade it. The difference between PROTAC and molecular glue, PROTAC people probably have heard a little more about because it's a little bit more established. PROTAC is linking two small molecules together with a linker. On the one end, it binds to E3 ligase. On the other, it binds to the target and then degrades. So it can be designed, but because it has two small molecules, it's usually challenging to have drug-like properties. Is relatively challenging. For molecular glue is a pure small molecule, so it overcomes the drug-like property issues. And also, a more important advantage is that it can target proteins that are previously undruggable, that doesn't have a binding pocket for inhibitor or for PROTAC. So this is the promise of molecular glue. For Degron, we have established a discovery platform to discover novel targets and novel degraders for molecular glue, this modality. So we have established very novel library of molecular glue degrader compounds of 10,000 compounds that are differentiated from other molecular glue degrader companies. We have 3 complementary screening strategies, and we have also established a very robust pipeline of first-in-class drugs. So Degron is oh, you can see this here. Sorry, I can see it over there. So Degron has labs in San Diego and Shanghai, and we are a member of J&J Labs, J Labs, the incubator, and we recently are also a Roche Accelerator member. We have raised 2 rounds of 35 million, and I'm in the middle of raising an A+ round of around 30 to 50 million. So we would be interested to talk to potential investors. So just a quick summary of our technology background, technology platform. So first, we have a very differentiated de novo designed and synthesized molecular glue library. This is based on our scientific founders' initial hypothesis that molecular glues bind to E3 ligase. Different structure of molecular glues create different new surfaces, and this will drive this to recruit different molecular glue targets. So we have created a glue library that has a different scaffold from other companies. All the molecular glue degraders initially were discovered by Celgene, and it was based on a lenalidomide core structure. And the glue companies we know so far, the published patents, they all used lenalidomide core. We use a different core that was discovered in our founder's lab initially, and we found a company based on that. Now we expanded to 60 different cores and 10,000 compounds that can be used for novel glue. So this just shows we have very good diversity, our library, and also very good drug-like property that meets the Lipinski rule of 5. And with this library, we use 3 complementary screening strategies. Because, as I said, molecular glues cannot be designed, so we have to have a library and to screen for novel targets. And we're the first company to use phenotypic screening and proteomic screening to discover targets in a target-agnostic way. So we've discover targets that people didn't know can be degraded by molecular glues. We also use, the third method we use is similar to what other companies use. We use an AI method to predict what proteins can be molecular glue substrates and do a reporter to do the screening. This shows how we developed our glue AI algorithm to predict which proteins can be degraded by molecular glues. On the left-hand side shows that there are 6 known molecular glue targets in the public space. And we have independently discovered and validated 7 novel molecular glue targets. So with the structures of these 13 novel protein molecular glue targets, we have developed an algorithm to predict what features are needed on the protein that can be degraded. So we predict about 3,800 proteins can be molecular glue degraders, and we can make reporter systems to screen our library. And we also have built our library, and we have done the 2D and 3D fingerprinting and AI analysis to quickly go from hit to lead to PCC to optimize our compound. And with that platform, we have generated a pipeline of programs in oncology, inflammation, and rare diseases. Our lead program is an RNA binding protein, which is previously an undruggable target. So we have taken this to preclinical development and plan to file IND in the 3rd quarter of next year. So there are companies who are doing glues, but we are different in that we can identify novel targets for the glues. So if you look at all the companies now, most companies are still doing molecular glues targeting IKZF1/3, which is the target of lenalidomide and pomalidomide. So they're making a different version of that degrader. we have only 4 companies have independently discovered and validated novel glue targets, and we're one of the 4. And the targets, because we are in a different chemical space, we can discover targets that are not discovered by the other 3 companies. So this is why we can discover novel degraders. I just want to give a couple of slides on our lead program to show how we do this. So our lead program, we haven't disclosed the target name because our patent is still being finalized. So we call it protein A for now. It's an RNA binding protein. It binds to the 3' UTR of pro-cancer survival, all the multiple pro-cancer survival RNAs, and stabilize these proteins, and stabilize these RNAs, and as a result, increase the expression of these proteins. We first validate what we found is a molecular glue by doing a few things, and we do this for all of our programs. In A, we show in the in vitro system that our molecule can trigger the polyubiquitination to show that it's a direct substrate of molecular glue, because a lot of companies discover something, but it turns out to be a downstream protein that's regulated by something else. So this is something, it's very important for us to show the direct substrate. In B, we show dose-dependent degradation of the protein. C, we show the ternary structure formation of a glue that is bringing together the target and the CRBN. In D, we show if we mutate the target protein A into a non-degradable protein, then the cell killing, we can abolish cell killing. And we're pursuing multiple indications of this very exciting target. And the lead indication is a BRAF mutant CRC with a high unmet need. We're also working on liver cancer, glioblastoma, and a rare disease called MPNST. So this just quickly to show our xenografts data. On the left-hand side, we show that the protein by itself can shrink tumor in a dose-dependent way in cell-based xenograft model on the top, and in patient-derived xenograft model in the bottom. And on the right-hand side, we show the synergistic effect of this protein together with BRAF inhibitors. So that's my presentation. Thank you very much for the opportunity.
Great job. So now, before you close out this part of the program, take out your phone. Everybody has a cell phone, mobile phone. What I challenge everyone to do is to raise the profile of these wonderful entrepreneurs, awardees, and speakers. Please, you all have some platform, whether it's Twitter, maybe X, LinkedIn, Doximity, Instagram, did I say TikTok, or just text to your students, colleagues, raise the profile, help these awardees and speakers to become the rock stars. They are the rock stars, but to be recognized as the rock stars in science. Thank you all.